everyone. We are so happy to have you, whether you are joining us here today or will be watching from the Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions you might have in the chat box below. We will have time to answer questions at the end of the presentation. My name is Monica Joanna Elmacave, and I am the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies and treatments for GABA-A variants by partnering with world-renowned researchers in GABA-A. We also strive to focus on the organization of patients for clinical trials. Today, we welcome Dr. Nathan Absalom, who recently joined us at the inaugural Cure GABA-A Scientific roundtable, which was held in conjunction with the American Epilepsy Society. Dr. Absalom is an expert in GABA-A receptor structure and function, with a large collaborative team at the University of Sydney, Florey Institute in Melbourne, and the Rikke Moller Danish Epilepsy Center. He aims to transform the diagnosis and treatment of genetic epilepsies associated with GABA-A receptor variants by unraveling the mechanisms underlying the unique circumstances of individual patients. He hopes that precision medicine strategies targeting the causes of the disorders can be developed. I'm handing it off to you, Dr. Absalom. Please share with us your presentation on natural history and gain and loss of function in GABA-A receptors.
Thank you, Monica. So I'm just sharing the screen. Can you see the PowerPoint presentation? Is that okay?
Yes. Perfect.
Yep. It's the correct one, not the presenter slide, hopefully?
It's the correct one.
Okay, that's great. Well, thank you so much for that introduction, Monica, and thank you for the opportunity to talk to patients, whether you're online or here in person. Thank you, or watching it later. Thank you for joining in. It's so important for us to be able to have these forums where we can communicate back to you the work that we do. We do a lot of work in GABRE receptor variants, and sometimes we get a little bit disconnected from the people we're trying to help, which isn't a good thing. So I think this is a fantastic initiative. And I really hope that this patient advocacy group, and I see that it's going to do very well, and I really hope it achieves the goals that it's going to achieve. So in saying that, again, thank you for logging in. I'm going to be talking about the importance of patient natural histories. I'm at Western Sydney University, so I'm the other side of the world. And sorry for the changes in time. We got a little bit confused with the time differences, and it sort of was at 3 in the morning. So yeah, we put it so it's a bit of a better time for me. But yeah, so going from where we start and why natural histories are really important. And I think the thing that we want to get across today is that the most important resources we have as scientists and clinicians is actually you. So when we had our first patient, the local hospital at the Children's Hospital of Westmead diagnosed, they came to us because we worked in GABRE receptors. And we'd spent a lot of time understanding how the receptors worked, but we hadn't really done that much clinical work, if any at all. And so we were talking about starting to move into this work, and the head of pediatric neurology was there at the meeting, and he gave me this advice. He said, the most important thing that you need to learn if you're going to do this, if you're going to work to translate your research into the clinic, is that you need to learn to listen to the patients. And at first I didn't really understand what that meant. But as we come along, I've realized that that was extremely important advice, and listening to the patients is absolutely the most important thing that we do. So let me give you an example of what I mean by that. So we have 2 patients that I've taken from the Cure GABA-A variant website, and there's a little bit of information about their disorder. And what we see is Pilar has a few different aspects to her disorder. So for instance, her seizures began when she was 9 months old, and they were initially diagnosed as febrile seizures. Then while she was growing up, they developed into non-febrile tonic-clonic seizures, and also has autistic features. Now, Gillian is a bit similar but different, and so she began having seizures at 3 months old. She has developmental delay and is non-verbal. She has hypertonia, and her seizures are uncontrolled and changed. And this is like typical of the stories we hear all the time, that each child is an individual and they have an individual course of the disorder, and it's very different. And so your experiences as caregivers, as parents, and the experiences of your children are all quite different to each other to some extent. But in saying that, there are some similarities between patients, and it's our job to try and understand what those similarities are, if they have a scientific basis, and if we can then use that knowledge to try and develop better treatments, or at least make the existing treatments work better for you. So the question that we had early on when we first had this child, which had a little bit of a different disorder and a very strange reaction to drugs, is we thought to ourselves, well, what if we go all around the world and we find as many patients as we can, and we listen to them. We listen to all their different experiences and try and figure out, well, which ones are the same, which ones are different, and then combine that with scientific research and understand whether there are scientific reasons for whether some patients have similar disorders and which develop in similar ways, and if there are reasons that some of these disorders are different with what appear to be quite unique aspects. So we had to do that was very difficult because in Sydney we don't see many patients, so we had to join up with experts internationally. And what we did is we incorporate this into a program. And so I think Anne is here, and she probably tell you that there's a very similar way that many of these rare disorders would be looked at. So we have a group, and we're trying to develop a precision medicine approach. That is our end goal. The precision medicine approach works with the patient basically at the centre, so everything's designed around the patient. And what we do is we go around and identify variants in GABA receptors, we determine their functional effect, and we take that knowledge back to the clinician, and then we compare the clinical data with the scientific data and see what matches. We can then, after that, start using that knowledge to look for drugs which might work on individuals. And we can also study representative disease models, but I won't be talking about that today. A little bit, but not much at all. So to give you an idea where we are at the moment, our clinical team in Denmark, that's headed by Professor Rikke and Moller, who's absolutely pleasure to work with, and one excellent scientist, the smartest scientist that you want to meet. She collects the data of every patient that she can find with a GABRA receptor variant. So she finds that through her clinical contacts, which are absolutely massive. And she also does it by looking through the literature and taking records from the literature and putting it into a database and trying to curate that database so it's as accurate as it possibly can be. Currently, it holds about 300 or more records of different patients. And what they then do is they then give back just the identity of the variant and nothing else. So we're kind of blinded to the clinical data, which is important. And we measure in Sydney what happens to that GABA receptor. So what changes the protein, how that protein is changed. And then after that, we bring it all back to the clinical data and see if differences in the functional response of GABA receptors can predict the clinical phenotype or correlated with the clinical phenotype, however you want to say it. So now that we've done that, and we've published some of our work, but over the next course of, I'd say, the next year and a half, most of the GABA receptor genes will publish similar stories on. And what we now know is that GABA receptor variants are at least two scientific problems. They're probably more, but the very basics is that there are two different types of variants. And some of you may have been told that you have either a loss or a gain of function variant. So I like to think of a loss of function variant on the left as basically like Wile E. Coyote when he's chasing the Roadrunner and he tries to light it and the rocket just doesn't work. So the GABA receptors have lost their function. And for those of you that do a lot of work reading up on the science of this, and I know in patient groups, some of the caregivers are just extraordinary how much knowledge they have about the science, and they actually tell us about papers that we should have read and haven't. But for those of you that do that, basically what we do is we incorporate the variant receptors into a cell system, then we apply different GABA concentrations, And if it takes more GABA to open the receptor, which is in the red, we call that a loss of function. So if the receptor, there's less electricity for the same amount of GABA. And if there's more electricity, the same amount of GABA, so if you don't really have to add much GABA at all, and then the receptor opens and there's a lot of chloride going through, we call that a gain of function. So those receptors are working more than they should. And if you have a loss of function variant, the typical diagnosis, and remember there's a typical diagnosis, so there will be some patients that are much, much worse than this, or maybe even milder than it. But typically it's associated with febrile seizures. Around 6 to 18 months is when you have your age of onset, you have autism spectrum disorders, intellectual disability, and tonic-clonic seizures as you develop. With the gain of function, you have a different set of phenotypes. So often you have focal seizures, but there's actually quite a lot different range of seizures that we get, that we see with the gain of function. The age of onset can be very young. We've had reports of mothers that have reported that they knew there was something wrong with their baby in utero and had had seizures in the first day of life. It was probably earlier than that. But typically the age of onset is 0 to 4 months. But again, there are some people that have it older than that. Almost everybody has severe intellectual disability. Many patients are nonverbal. And for a section of patients, they have some movement disorders, which are very, very debilitating and quite difficult to manage. On the right, you can see this clear segregation, again, for those of you more mathematically minded. We have these two populations with very different ages of onset. So we know that. So we know that this is a disorder where there's at least two categories that will probably need two different treatments or at least plans. So if we go back to what the patients are telling us, you can see, and Pillar and Gillian have already been obviously diagnosed, one with a loss of function, one with a gain of function. And so we can start to look at their phenotypes and tell a little bit, look at the progression of the disorder and start to tell a little bit more about what's happening to them by looking at other patients with similar variants. So that's really the power of what we're trying to do. So at the moment, we've tested about 250 variants so far. And one of the things that we want to do is give you answers. I think that, our experience when we first started this project and GABRA receptor variants were starting to be identified in these patients, the clinicians would tell us that one of the things that the caregivers and patients are not liking is the lack of answers. That a lot of the questions, they couldn't give answers, very fundamental questions of what you need, what's going to happen, how is this going to progress? And there was very limited information. So we've really tried to put better information into the hands of both caregivers and to clinicians. So there's a lot more, not certainty, but you have a lot more, your expectations are closer to reality, I think, when you get diagnosed. So we published this last year, and there's a little guide. So when you have an identification, in this case, a GABRB3 gene, there are some key features for the clinician to look for. And then we state some of the features that you'll develop as you get older. And that's typically what we're going to be showing in the next year or so for all the different GABRA receptor subtypes. So you'll have some idea, what is going to happen. And here you can see some of the different variants, some different patients, again, on the website. And if you follow the little guide that we have, you can see whether they're going to be loss, gain, or loss, just looking at the phenotype and looking at the box. And that's important because it takes a bit of time to do functional testing. So it is quite nice if from simply the clinical data and having the genetic test, then you've got a likely course. So the benefits of what we're doing, the benefits of getting all this information from you, is that we'll have more accurate knowledge for families of how this disorder is likely to progress. What we can then do is we can understand the consequences basically of the receptor tool. And we can start to design prediction tools based on the variant identity. I'll talk a little bit about that in a minute. Another thing that we'd like to know, and I think this is like quite important, is to find the most important part of the disorder to treat. So as I said, different families have different, each child is different and there are individual problems. And for some people, some aspects of the disorder are much more important to treat than others. But that's not actually how we're evaluating treatments at the moment, which is not a good thing. So we want to move to being able to evaluate treatment regimes on a like-to-like basis. And what we want to do is define cohorts that are appropriate for clinical interventions. And that's really critical because we don't want to throw out a good drug used for the wrong disease because we put patients together that simply aren't the same in a scientific manner. And therefore the drug works differently on the different populations, has an adverse effect on one set, and then clinicians become scared to use the drug. And that's something we really don't want. And that is a genuine problem. So as I said, the first patient that we looked at was this child at Westmead Hospital. And the reason we were looking at it is because it had a very, very odd reaction, adverse reaction to a drug called vigabatrin. Now, it's quite logical to use a drug like vigabatrin because what it does is it increases GABRG activity indirectly. Basically what it does, it stops GABA being broken down in the cell, so there's more GABA released. So your receptors can detect it and activate it. So we know that there was a patient with a truncation variant that Rikke had not treated, but had been involved with, where if you gave the patient vigabatrin, it was seizure-free. So it was very successful, it restored the function. In that mild loss of function case, it's a very good treatment. But the patient that we were looking at Westmead was a gain of function. And what it did is it exacerbated the gain of function, so it released more GABA, more of the receptors open, more of the receptors open than it should, and this caused a severe catatonic response. And when we started chasing around, because Rikke was starting to build this database, she found another patient in the UK that had exactly the same response, and it also had a gain of function. And so we published that. And then after we published it, we started to get a few emails from other clinicians who hadn't recorded that adverse reaction, but had that adverse reaction. So now you're unlikely to get vigabatrin if you have a gain of function in these variants. And that's a good thing. It's not a nice adverse reaction to have. But this is really important because what we need to do is be able to trial therapies on the correct patients. So by stratifying patients based on the functional change in the variant, and then looking at what the therapy we're actually trying to develop is, because that's quite important, we can then decide whether whether it's appropriate to put the patient in the clinical trial for that drug. And as I said before, that will help us to get the best drugs to you. So essentially, what I'm saying is we really require a precision medicine approach that's targeting loss or gain of functions. So what we want to do with loss of function is we want to target a treatment that's targeted increasing GABRA receptor activity. It's a little bit more complicated than that because we have lots of drugs that do that. Benzodiazepines do it, for instance. So Frisium, I think it's a brand name of clobazam. Phenobarbital, there's a lot of common drugs that do that, but they also have side effects that are not nice and are not appropriate for everybody. Unfortunately, for the gain of functions, there aren't any treatments targeted at decreasing GABRA receptor activity. It was generally thought that that would be poisonous and deleterious. So drug companies, as a general rule, if they'd identified a drug that decreased GABRA receptor activity, they would have discontinued that drug very, very early on in their screens. So that's not very good. It means there aren't a great deal of drug candidates that we can use or look for. So moving on to the natural history studies and what I'm sort of advertising, I suppose, here today and why they're good. The thing is that natural studies can tell us things that we don't already know. So what I've explained to you is the information that we've been able to get using direct clinical sources and indirect literature sources. Now these are really good, for various things, but not so good for other things. So we have direct contact with clinicians, and what happens, the information gets collated into the database, and it's given in standardized terms. Now, this is really useful for us nerds. So when we want to do statistical analysis, it's great. Having standardized terms is very, very useful for that. But it's not a really good description of your child. Right? It's fitting your child's disorder into a little box, into a little term that's useful for us. That doesn't necessarily mean it's that useful for you. The other problem is that it becomes difficult to update. So if something happens, and it's something important for us to know in the development of your child's disorder, it takes time for that information to come to us. And we've already found the limitations of that. So we have a paper that we published recently. What happened is we made some predictions based on our functional analysis of how severe the disorder would be. And when we looked at the clinical data, we noticed that 1 or 2 patients at the more severe end had movement disorders and epilepsy of infancy and migrating focal seizures, which are very severe cases. But it wasn't clear that that was associated with the functional change. So what Rikke did is she went around every clinician that had uploaded that data, and then found actually what had happened is that a lot more of those patients in the severe case had the same thing, had the same EIMFS, and they had the movement disorders, but they didn't necessarily know that when the data was uploaded to the database, and some of the disorders weren't, there wasn't a box in the form. They basically filled in the standardized form, and some of them were writing it in and some of them weren't. So it's important to be able to update it and have really good relationships with the people who are entering the data. And unfortunately, clinicians, as you know, are very overworked, and managing databases is not always their thing. And also on treatment response. So when we get data on treatment response, it's recorded simply as what the percentage reduction in seizures are. Now, seizure reduction is important. Obviously, it's important, but it's not the be-all and end-all of treatment. It's not a quality of life measure. And so I find that that's probably the biggest limitation in the data that we have so far. And it's that we get very poor information on the response to treatment. The indirect literature sources are useful to a point because they allow increased rigor for statistical analysis. So they're really good at increasing the numbers, but they're even worse than the direct clinical source because we can't go back and ask them any questions. We don't know who the treating physician is. And sometimes we try and trace authors, and it's very difficult. So the information here is relatively limited. However, when we do natural histories, it's a little bit different. We can have direct contact with families. We can have direct contact with caregivers to give us the information, and that information becomes unfiltered. So what you're telling us is what you observe directly. It's not information you give and then somebody else says, oh, okay, well, this goes in this term and off it goes to the database. We always have access and can go back to that. The next thing that's really important is we start to learn what's important to the families. So, as I said before, we know that seizure reduction is important, and we know it's important because that's what the FDA wants. But that doesn't necessarily mean that that's the most important thing for families, because often it's little things as well. It will be, this is nice to stop my seizures, but my child is not sleeping through the night. It sleeps for maybe half an hour at a time. Is there something you can do for that? And there can be other things that are quite traumatic. You can have very, very painful dystonia, these very, very painful muscle cramps, for instance. And just because you get on top of the seizures doesn't necessarily prevent that particular painful part of the disorder to stop. So it helps us think about what treatment that you really need, that some of your patients will really need, and how we should be measuring that. So I think that's a really important thing, that we get out of natural history studies that we don't necessarily get out talking to the clinicians. Well, sometimes we do, but not always. Because there's better updates, we get a better idea of the disease progression, and from that we'll be able to predict. And I think that's really important, knowing the disease progression. Our experience is, it's not just that the individual patients are different, but because you're all over the world, you interact with different healthcare systems, and those healthcare systems have different quirks. So knowing what you need, what tests you have to arrange, what specialists that you might have to see as early as possible. We've had some people mention that that's what they really want. We can get, as I said before a few times, with natural histories, we get a better description of treatment outcomes. So instead of just saying, this is a reduction in seizures, somebody can say, yeah, not only, okay, the seizures didn't reduce that much, but my child has autism and a lot of the tantrums have ceased. They're interacting with other kids, which they don't want to do. They're able to go to school. That's a real positive that we wouldn't necessarily record if we just looked at seizures, because they're not always correlated so closely. And so that's really important for us to know. And when we do that with what we call deep phenotyping, which is essentially when we employ somebody, talk about who we employ to do that, where we employ somebody to really go into the data and find these things out, then we're going to have better answers to your questions, and hopefully, as we say, can move forward into better treatments, which is what we all want. So, as I sort of alluded to, we call it epilepsy. So we call these epilepsy-associated variants because seizures are common to pretty much everybody. Although there are a few patients that don't have seizures. But that's not really a description of the disorders. I think you all know that's not what we're dealing with on a day-to-day basis. And there are other parts of the disorder that have substantial impacts on the family. So I think maybe 20% of patients we find have movement disorders that are severe, and the children are non-ambulant. So this can also come with painful and uncontrolled muscle spasms. Now, there are surgical options available, which I won't talk about so much today, but that's, a difficult conversation, and knowing what's likely to happen and what are the benefits and risks of those surgeries are important, and we're trying to get on top of that with some more information. And often for those patients, little things mean a lot. Being able to control movements in, some very severe children, movements in the face and eyelids, and being able to smile and sleep is like huge. Intellectual disability is quite problematic because the majority of patients will have some form of intellectual disability. In fact, the vast majority will. A substantial proportion of these are severe, non-verbal, but it's when it comes to treatment. So they may require early intervention, and looking at when treatments have been given and their utility and how they assist patients with intellectual disability, I think is really important. And again, that's information we just don't have. Do these particular drugs, these treatments that you have, are they improving the development delay of achievement? A small proportion of children have very, very severe disorders. They have feeding difficulties, they have to be fed by tube, and the risk of early mortality is very high there. A lot of other patients as well have, as I mentioned, neuropsychiatric disorders. And for some families, we've been told that this really is the major problem that they have, because the autism spectrum disorder will be very severe in those patients. And we're already seeing, if we use personalised approaches to tackling the cause, they're likely to be superior to generic treatments. I can give an example of that a bit later on. So again, if we go and we look at one of the patients and bring it back to you, as it were, it's thinking about what has the most impact on daily life. So Charles has a variant in the alpha-2 receptor. He's non-ambulatory. So we start to think of ourselves, well, with this movement disorder, does he have painful and uncontrolled muscle spasms? For the whole patient cohort, when do these movement disorders begin? How do we detect them? How do they progress? How are they affected by the drug therapies that you're getting? What are the surgical options and what are the risks? When do we get help? When is a good time to see a movement disorder specialist? And what do they do which can help? And can we predict what's going to happen with our knowledge from the variants. So when you first get diagnosed, can we give you a good idea of whether this is going to be a problem? Maybe you live in a, and simple practical things, isn't it? So maybe you live in a house that's 3 stories and is small and has lots of stairs, and it's just not going to work. And you want to make plans for the future. Again, Charles has global development delays, non-verbal, and this really is the majority of patients won't necessarily be non-verbal, but the majority will have some form of intellectual disability. And we start to think, well, is early intervention going to be required? Are any of these anti-seizure medications that we take improve cognition, or are they separate? And if we can get on top of the seizures, will my child be able to talk about what's going to happen to my child if we can get on top of the seizures? And all the issues that go with it. So here's a really good example where when we target GABRA receptors directly, we can get better quality of life measures. So this drug, vinpocetine, it's not available in every country. I think in Australia it's not available, I know that. But this is one example, and it was used in a few patients with GABRA receptor alpha 1 loss of function variants. And it increases GABRG activity. So that's why it was trialled. And what happened is, you can see from the title exactly what happened. The vinpocetine improved neuropsychiatric and epileptic outcomes. But the most important thing that was happening to that child was the neuropsychiatric, the autism spectrum disorder, was very, very well managed. It was very obvious to anybody who interacted with him when we started this therapy, and based on his behavioural difficulties alone, and a lot of that was ameliorated. It also improved the seizures, but the effect wasn't quite as dramatic. So when we have natural history studies, they allow us to evaluate the quality of life improvements of different treatments without us having to do a clinical trial. So we don't have to, we don't do the follow-up. So yeah, the follow-up for clinical trials obviously is a lot more involved, and natural history studies allows us to sort of do that in a slightly different way. But with these existing treatments, and you'd probably be aware that there's lots of different things that get tried. And when you talk to other parents or other caregivers in these patient advocacy groups, you'll start realising that a lot of different drugs are given for seemingly no rhyme or reason. And when we look at the data as a whole, we actually find lots of drugs are given, but there's not really any good information that we can use to give advice. So families can give these evaluations of treatments from a different perspective to the specialists, and I think that's important. So a clinician, as I say, sometimes we just measure seizure outcomes, but you can tell us exactly what the benefits are on a quality of life basis. Now, in saying that, the FDA require a measurement of seizure burden, and that's probably why it's always recorded, and clinicians are very, very thorough on measuring that. So it's a necessary requirement, but I don't think it's a sufficient requirement for a good treatment. Cool. So some of the other things we can do for natural history studies are better predictions. So here's an example of what we can do now. So this hopefully is going to come out in brain soon. But what we can do is we're starting to get enough information so that we don't have to, we're out of a job, right? So we're starting to get enough information from the work we've done that we can bypass the functional work that we do in Sydney, and we can make predictions based on the variant and the structure alone. And it's really quite simple. These receptors work by GABA binding at one spot and an ion channel opening at another. We can just draw a straight line between the two spots, measure the distance from where your variant is from that line, and that gives us a really good indication of whether your variant is pathogenic or not. Because there'll be some people that just by chance have a variant in GABRA receptor which doesn't actually do anything, and they have a different cause. And so we're pretty good at just being able to do that by an algorithm. We're not quite so good yet at being able to compare gain versus loss of function, but we're reasonably good. We can make reasonably good predictions. And if we just move beyond the simple structure and go into a little bit of clinical detail, then we make very accurate positions. But we have no ability to predict what treatments are best. We can't do that, and that's what you want. What we also can't do is we can't predict how mild or severe your disorder is going to be, and we can't predict the progression of the disease. So we want to be able to do that, and we need a lot more data to be able to do that. And here's an example of where they can do it for SCN1A genes. So SCN1A is like a much bigger gene. They have a much bigger population that they can determine. And our collaborators is in Texas now. But our collaborators in Texas developed this algorithm where you can just put in the age of onset and your gene variant, and then it tells you your probability of developing either febrile seizures, which are limited and you have very little intellectual disability, or you have Dravet syndrome where it develops these tonic-clonic seizures. There's a high risk of status epilepticus and death and developmental delay and a lot of other associated co-occurrences with the disorder. And this prediction tool gives a fairly accurate prediction of how that disorder is going to go. And we want to be able to do that for GABRE receptor variants. And we need to know more from you about what happens to be able to do that. We want to be able to predict specific aspects of the disorder. So we want to be able to predict whether your child's going to be nonverbal or non-ambulant. And again, we just need a bit more data. I don't think we're that far from it, but we need a little bit more specifics to be able to do that. Again, we want to be able to predict how severe the intellectual disability is likely to be, what you can expect from different drug treatments, and how much assistance in terms of care are you going to need. And again, that's important in different countries. So I know in Australia, we've just completely reorganised the way we give care. So we have a national disability insurance scheme, for instance, and that's in flux as to who gets money and what money people are given. And knowing what assistance in terms of care you need, I think can be helpful in the political side at times as well. Just putting it sort of as gently as I can. So on the specifics of it, let's say I'm a scientist, so I do the basic research, and it's nice to be able to talk to a patient group, even though a lot of you are listening, because they basically tell me, no, you're not allowed to talk to patients. No, you're the scientist, you don't have the experience. The natural history study is likely going to be organised through Rikke. So she's got extensive experience in rare genetic epilepsies. She's got access to large international expertise, and it won't be like reinventing the wheel. And she can separate new and existing data sets that are crucial for these predicting. So what we can do is we can either say we need more numbers, we can put data sets together to get more numbers, or we can separate them, and that allows us to work with one data set and then say, okay, we're making predictions in that data set. Does that hold in another data set? So that's actually really quite useful for us to have it sort of separate. But these are discussions into exactly how it's going to go. So I only analyze the data, and Dr. Sebastian Ortiz, he's doing the deep phenotyping, so he's very interested in very fine details of what's happening with the children and developing it into a model. So you're likely to hear from him as well. So that's great. So this is everybody that's involved. It is a really big collaboration, and these are my colleagues at Sydney Uni. So Philip, Mary, Vivian and Susan have done a huge amount of work. These are the people that fund it. And Katrine, Rikke and Sebastian are those in Denmark. So thank you for that. I'm quite happy to answer any questions. And we've still got time, that's good. I did deliberately have it a little bit shorter than the hour. So I'll end the slideshow and stop share. Cool. So thank you. Oh, and thank you for that knowledge, Augustina. So top room, I'm actually I don't think I've had. I'll have a look at. I'm not on top of the Alpha 1 or the A1 data set, unfortunately. I'm more on top of the beta 3. I'll pass that on to Vivian, who's definitely Verapamil. Again, I'm probably not willing to say too much on Verapamil. I haven't seen I haven't seen too much hard data on Verapamil. What I might do is, afterwards, if you want to just give me your email, I can go back to Vivian and just look a bit more onto your specific variant, because I'm pretty sure it would have been tested by now. I think some of these possible treatments, we're always a little bit more cynical. Cool, thanks for that. We're always, I wouldn't say more cynical, but we always like to see how it works in different systems. So I think Verapamil was one of those. I'm not sure if it was one that increased GABA activity, increased the receptors at the cell surface or changed the function. So what's kind of important is with your variants is some of them are going to, particularly the loss of function, some of them are going to reduce the amount of receptors at the cell surface, and that happens for deletion and truncation variants. And some of the missense ones will do that as well, but a lot of the missense ones actually change the ability of the receptor to act when it's at the cell surface. For those particular variants, it's much more likely that you need a drug that targets the receptor directly, because even if you get more receptors at the cell surface, they won't work, basically. So I'll have to just have a look. My impression is that Verapamil was proposed to increase the receptors at the cell surface, but I'm not sure what that particular variant does.
Sorry about that. I just got taken out because my internet is in and out. So I apologize for coming in and out that way. But I wanted to say thank you so much for explaining the importance of collecting data from our patients. We really needed to understand how we can create prediction tools and ultimately have a better understanding of how these drugs work based on gain, loss, and gene. We have a link in our website called Get Started. That's on cureGABA-A.com.org. So we're really encouraging everyone to go register, join, submit, participate, please, so that we can get our community's information to researchers like yourself. We also have a link where you can request a one-on-one appointment with Dr. Rikke Moller and Sebastian Ortiz via Zoom. And I know that you started asking some questions, answering some questions and comments. Did you answer Augustina's comment about topiramate?
Yeah. Okay, well, I took the details, so I'll have a look.
Great. I also wanted to just piggyback off that. We have a really great tool that was just presented to us recently called the Variant app. And the Variant app is another data collector, and it helps us to record the drugs that our community is taking and also the effects of those drugs. I have a question here from Lisa. She was asking whether alpha-1 mutation is most likely a gain or loss of function. And I know that you went over those slides, and it's just another reason why we need to share more data on our kids, like the key features, supporting features, developing features, so that we can understand whether children are gain or loss of function.
Yeah, so it's actually a good question. So the way the receptor works, it's not, each gene is not equivalent. So the beta 3, for instance, the betas in the receptor, they do a lot of the legwork in opening the receptor. So variance in the beta genes, we actually see about a 50-50 split with gain and loss of function variance. In the gamma-2, that is more likely to, what it does is it gets the receptor to the cell surface. It doesn't actually bind to the GABA. So there's a great deal fewer gain of functions in the gamma, for instance. And the alpha, it's a similar issue. So it doesn't make quite the same, do quite the same amount of work to open the receptor. So there's generally a lot more loss of function variance in the alpha one. In saying that, we still find gain of function variance in the alpha one, and they're not quite as severe as the ones that you'll see in the beta 2 or the beta 3. Again, I think the best thing to do is to set up an interview with Rikke and Sebastian. And if they don't know, if they haven't tested it, then they'll put it in for testing. So we'll actually test it for you. And if it has been tested, and quite a lot of them have been tested, then they'll either know and can tell you, or they can contact us and we can tell you.
Amazing. Great. I'm just going to take a look and see if there's any more questions here. Cora is saying, you mentioned that there may be different factors that may be the most important for treating each patient, such as autism. Not to open a can of worms with a controversial topic, but one of the mainstay treatments for autism is behavioral therapy, such as ABA, which is very controversial. Some autistic adults claiming it is abusive. Given the genetic causes of autism in the GABA kids, would that be a viable treatment option, and should that be explored?
Yeah, so that's quite a good question when you start getting away from treatments that are directly targeting the receptors. I think, my advice would be, and I think, one of the problems here that you have is that you will be going to a specialist, or you'll be talking to specialists that deal with children that have autism from a range of different causes. And because they have a range of different causes, they're going to respond to different treatments, even if they're not drug treatments, in different ways. So, essentially, I can't answer that question because we've not taken the data. We don't know, because we don't know what patients with GABA receptor variants have tried this and whether it's worked or whether it hasn't. And so what I'd say is that, unfortunately, I can't give you a good answer to the question, but that's really why we're doing what we're doing, so we can give you that answer. I hope that makes sense. I'm sorry I can't give you more, a better answer.
What I'm hearing is the echoing of we need more data.
Yeah,
we need more data.
Right.
And if we could just encourage all of the caregivers to go to the website, curegaba.org, and go under the tab, Get Started. There's 1, 2, 3, 4, 5, 6, and it's going to continue to go. Follow the steps and just please participate. If there's no more questions, I just wanted to really thank you for being here with us today, taking the time out of your day to be with us and explain everything that you have so clearly. We really appreciate everything that you're doing for the GABA-A community.